Hey everybody, we are on our last unit for lab here, and we're doing our first lab video on muscle. So we're actually going to look at three types of muscle tissue today. There's skeletal, cardiac, and smooth, and that is in order of deliciousness. Um, Skeletal muscle, of course, as its name implies, is attached to bone, it moves bone. As we're going to see, skeletal muscle, the cells are these long, skinny fibers. In fact, they're called fibers because of their length and their skinniness. We're going to see that skeletal muscle is striped or striated, as well as being multinucleate, meaning each cell, each fiber, has many nuclei. And of course, skeletal muscle, for the most part, is voluntary, except for reflexes and things like that. So we're going to look at skeletal muscle today. We're also going to look at cardiac muscle, which is in the heart wall. Of course, it's involved in pumping blood. Now, no long, skinny fibers here. Instead, they're short and they're branching. They will have the striations or stripes, but only 1 to 2 nuclei per cell. And one thing we're going to see in cardiac muscle that's characteristic only of cardiac muscle are these things called intercalated discs. And of course, cardiac muscle is involuntary. You cannot, tell your heart voluntarily to beat stronger or to beat faster. Finally, we got smooth muscle. So we're going to find that mostly in the walls of hollow organs. So think about like blood vessels, intestines, stomach, esophagus, ureters, uterus, urinary bladders, those sorts of things. And smooth muscle is involved in moving material through those hollow organs. The cells in smooth muscle are described as being fusiform in their shape, which basically means that the cell is kind of big in the middle and then it tapers and narrows at each end. And I drew one nucleus in this cell because these guys are uninucleate. They typically have one nucleus. And there are no stripes, no striations. In fact, that's where we get the word smooth, because there are no stripes, no striations. And of course, smooth muscle is involuntary. Okay? All right, so those are just some facts about my three types. Let's actually go ahead and take a look at all three, starting with skeletal muscle. And I've got about, let's see, 1, 2, 3, 6, 8-ish skeletal muscle fibers going horizontally here. Let's see, let's use blue. So this is, one skeletal muscle fiber right here. It's actually much longer than this. It's been cut on this side and on this side as well. So this is one skeletal muscle fiber here. I do see lots of nuclei, right? Every one of these fibers has a bunch of nuclei. That is why we say they are multinucleate. All right, what else do I see? I can totally see the stripes. Can you see the stripes? So I got these alternating bands of dark pink, light pink, dark pink, light pink, dark pink, light pink, etc. And this pattern that I'm seeing is because of the way the contractile proteins are arranged. And there are two contractile proteins we care about. One is called actin and one is called myosin. We'll get more about those in lecture, but for lab, we're going to know that these two contractile proteins exist. And of course, contractile proteins contract, right? Powerfully cause the muscle cell to powerfully shorten. All right, let's take a look at another one here. So tell me how many fibers you see here. Make sure you see striations. Make sure you see the multiple nuclei per fiber. And with that, let's jump into the histology of cardiac muscle. So cardiac muscle cells are, of course, in the heart, right? Helping to pump blood. They are not known as fibers because they are short and they branch. Like I can see some branching going on right here. See that branching going on right there? I see some branching going on right there. The branching is actually going to help signals spread from cell to cell to cell. Like the more branches you have, the easier it is for signals to move between cells. So most of my cells are uninucleate, like one nucleus per cell. And what I also got to notice is I do have striations. They're just like, they're fainter. I mean, if I look right, here, I can see all these little lines in here, right? If I look down here or right here, I see all these little lines, all these little lines here. Those are striations. So they're there. They're just not as, prominent. They don't like jump out at me the way they do in skeletal muscle. Okay, I've also got these structures called intercalated discs. And you can think about the word intercalated as meaning stuck in between. So intercalated discs are in between cardiac muscle cells. So like this line right here, this red line is an intercalated disc. Here's another one. Here's another one. Once you see one, once you can find one, you should be able to find just a whole bunch. And every time I see an intercalated disc, that means there's a cardiac muscle cell on one side of it and another cardiac muscle cell on the other side of it. And because they're in between cardiac muscle cells, it's going to make sense that their job is to join them together. And they actually join them together in two ways. They do it mechanically via structures we know called desmosomes, and they also do it electrically via structures called gap junctions. I got millions of cardiac muscle cells. I want them to contract and contract powerfully. I don't want them to pull apart, so I need them to be anchored to one another with desmosomes. I also want them to have a coordinated contraction. So I need electrical signals, depolarization, to spread from cell to cell to cell. And that is why I've got gap junctions. All right, so we got some nice cardiac muscle here. Let's look at another one. So you should be able to find me a ton of intercalated discs, faint striations, nuclei, and be able to recognize this as cardiac muscle. Okay, last muscle tissue of the three for histology here is smooth muscle. So these are smooth muscle cells here. They're running vertically. Over here, I've got smooth muscle cells that are cut transversely. Notice I got no stripes. I got no stripes going here. That's why it's smooth. These are non-striated cells. They still have contractile proteins. They just don't have that pattern that we see in skeletal or cardiac muscle. And of course, remember, we've got these smooth muscle cells primarily in the walls of hollow organs, arteries, esophagi, uterine tubes, etc. Another picture for you. Smooth muscle cells running kind of like diagonally here, and here they're cut in cross sections that are coming out at us. And yeah, that's it. Smooth muscle, kind of boring under the microscope, but easy to recognize. All right, what we're going to do now is shift and look at a model. This is a model of a single skeletal muscle fiber. Remember, a skeletal muscle fiber is a skeletal muscle cell. They're just long and skinny. And I see the plasma membrane right here. The plasma membrane of the skeletal muscle cell is called the sarcolemma. Remember, we had an axolemma and a neurolemma in neurons. So now we got a sarcolemma for my muscle cell. The word sarco means flesh or meat. So kind of makes sense. Now around my sarcolemma, I see a layer of loose connective tissue, which is called the endomysium. This is kind of like the equivalent of the endoneurium that I had around an axon. The endomysium is going to attach neighboring muscle cells to one another and also give me a connective tissue structure that the muscle cell can pull on when it contracts. Okay, so we got a couple things in this view. Let's keep on keeping on here. Turn this guy around a little bit. So I still see my sarcolemma here. This is my plasma membrane and my muscle fiber. I still see my endomysium, but now I see a motor neuron here, okay? I see a motor neuron, all right? This is a somatic motor neuron right here. These are some telodendria of it, and this is its axon terminal or synaptic knob. So in here I would find, synaptic vesicles with neurotransmitters that are going to be released, and they're going to stimulate this muscle cell to ultimately contract. So telodendrion, axon terminal, and right here they cut the axon terminal away to show me the part of the sarcolemma that's synapsing with the axon terminal. And this portion right here, that part of the sarcolemma looks kind of like a seashell or a leaf. That is called the motor end plate. And the motor end plate, notice that it has like folds to it. The folds give it more surface area. More surface area means I can have more receptors for the neurotransmitters that are released from the axon terminal. Okay, excellent. Let's spin this guy around again. So in this view, I can still see my endomysium. I can still see my sarcolemma. I actually see an axon terminal or synaptic knob cut right here. And I see the motor end plate, and look at all the little folds it has. So synaptic vesicles in here, releasing neurotransmitters, binding to receptors, and I maximize the number of receptors by maximizing my surface area by having folds. Now, what else do I see? I do see a nice nucleus over here. And of course, there would be many because this is a skeletal muscle fiber, which means it is multinucleate. But now, interestingly, I see all these cylinders in here. Now, these cylinders are called myofibrils. Notice that in the cylinders, I got dark pink, light pink, dark pink, light pink. So these are also responsible for the stripes, for the striations that I saw earlier. Now, the myofibrils are primarily made of contractile proteins, including the two we mentioned earlier, actin and myosin. And so myofibrils are very much involved in the process of muscle contraction. All right. So myofibrils, endomysium, sarcolemma, motor synaptic knob or axon terminal, motor end plate. So lots of good stuff in this view. I think we're going to spin this guy one more time. Look at it from the top. And I see my sarcolemma there. I see a nucleus. And I see just a bunch of myofibrils in cross-section here. And that's my last view of this model. And finally, we're going to switch up to some histology and put some things together. So this is skeletal muscle. Again, look, I got 1, 2, 3, 4, 5, 6 skeletal muscle fibers running horizontally. Note their striations. I actually don't see their nuclei very well here, but I know they're going to have multiple nuclei. But what I do see here are synaptic knobs, axon terminals, right? I see axon terminals here. So I got telodendria. The black lines here are telodendria. So what I'm seeing here is actual motor neurons synapsing with skeletal muscle fibers. If I were to rip off this axon terminal here, this synaptic knob, if I were to pick it up, I would see the motor end plate underneath it. And this synapse here, between my motor neuron and my skeletal muscle fiber, that synapse is called a neuromuscular junction, obviously because it's where a neuron is meeting a muscle cell. And that is it for this first lab on muscle. That was really short. That's okay though. Next one will be longer, don't worry. And I'll see you then. Bye-bye.